Thank you very much for your interest in the Protein Maker from Protein Biosolutions. This is our workhorse for high throughput parallel purification. The video here is designed to give you a bit more of an in-depth look at the instrument that is used to accelerate purification processes and reduce risk of project failure in our customers' labs. This video should be able to serve as a virtual demo for any of you who are unable to visit us or a nearby location to see the instrument in person. The Protein Maker has been designed for fully automated parallel purification of up to 24 proteins in a single run. There are 24 independent channels on the instrument, each driven by its own pump. You can see 12 of those pumps here on the front of the machine, and there are 12 more that you'll see in the back of the machine. Since the software provides individual control over each channel, in many ways the Protein Maker fulfills the role of 24 separate FPLCs, but with some additional benefits. For example, it fits in a much smaller lab footprint than 24 separate instruments would, and requires only one setup and not 24 separate setups, which would be quite time consuming. These and other benefits enable our customers to take good advantage of the parallelized approach to purification and arrive at the project's end goal much faster. Let me give a couple of application examples. One is risk-free scale-up, which enables us to scale up a successful purification run that yielded, say, 20 milligrams of protein to a run that yields 100 milligrams or more of protein simply by using 5 or 6 of the 24 lines all at the same time. In this example, 6 parallel lines yielded 176 milligrams of protein when we only needed 100 milligrams for the project. And we were able to do this quickly and easily in one run with a purification protocol that we knew was going to work. A second example is what we call lysis scouting. This is a good example of a very useful experiment that would likely not even be done if our users did not have access to the Protein Maker. From a 1 liter prep, we will pull out about 30 milliliter aliquots and lyse the cells using 12 different lysis buffers. Then in a 30 to 40 minute run on the Protein Maker, we can process all 12 samples in parallel in order to determine which of the lysis conditions provides the best result. Sometimes the results are dramatic, as in this case, where only 2 of the 12 lysis conditions were successful. By doing this and other parallel experiments as a matter of routine, our users are able to eliminate a lot of the risk associated with their projects. Well, there are many other case studies and other application information available. Feel free to contact us if you'd like to see it, or check out our website. Look for the slide deck that's posted on the website. But with that general introduction, I'd like to walk through more of the specifics about the machine itself, and then I'll actually have a demonstration experiment that we'll go through in detail as well. Let's start by taking a closer look at one of the 24 channels on the Protein Maker. Each channel on the instrument is composed of a syringe pump, a syringe, a 9-port valve, and the associated tubing and connectors as you see in this picture. So in this picture there's a 5 milliliter syringe, that's the glass syringe in the bottom, and other syringe sizes are available from as small as half a mil or a mil up to 25 mil syringes are available for the Protein Maker. Each of the channels on the machine is completely separate from the remaining 23 channels, so each protein really has its own separate flow path, and there's no real opportunity for cross-contamination between the lines. The schematic here provides a good overview of one of the flow paths on the instrument. While this represents one of many possible configurations, it should give you a good sense of the options available for configuring the Protein Maker. In this case, each line has access to a protein sample, several different buffers, a waste container, water for cleaning, the column where the protein will ultimately be loaded and purified, of course, a needle to aspirate sample up from one of the positions on the fraction collection deck, and a step or passive mixer that can be used to create mixtures of buffers to be flowed over the column. For example, to create a sample of 50% elution buffer, you can load 0.5 mils of elution buffer in through the syringe and 0.5 mils of equilibration buffer in through the syringe before pushing it over the column. We'll see that in the demo experiment here in a few minutes. This is just one configuration of many that are available, and each user can decide how to most efficiently configure the lines for their own processes. And again, this is just one of the 24 channels on the Protein Maker, so there are lots of options available. Next, we're going to take a look at the area where the columns are typically connected to the protein maker. The tubings that lead to the column and any needles that are set up to aspirate from the fraction collection deck lead here to the gantry pads in the back left of this picture. Now in this picture you can see a few columns are connected and organized here on the gantry pad. Gantry pads each contain 24 positions that are formatted to be compatible with the standard 24-well SBS plates. In this picture we have a few 1 mil and a few 5 mil columns attached, though larger or smaller columns could be used as well. There are two gantry pads shown in the picture on this protein maker, which provides the user with options to use some of the positions to aspirate samples from the fraction collection deck or to allow for automated multi-column purification experiments. The gantry pads are connected to the XYZ gantry, which is that post in the back, which has the ability to move in the X, Y, and Z directions as defined by the user. Zooming out again, we see the entire fraction collection deck that is accessible by the XYZ gantry. The deck itself has 20 different positions, with one of those 20 used as the waste position there on the left side. Each of these deck positions can hold an SBS formatted plate or a set of tubes, like the one shown here. So 19 positions really are available for collecting fractions, collecting flow-throughs, washes, for aspirating a fraction from the deck, etc. Additionally, there is an option for this to be a chill deck that is actively cooled through a low temperature circulator through a pipe that goes through the deck itself. However, if you have a cold room and you prefer to place the entire instrument in a cold room, that works as well. We have users who store the protein maker in its entirety in a cold room full time, and that works just fine. Just to give you a sense of scale, the instrument itself is 44 inches wide, 30 inches deep, and 44 inches tall. So it's bigger than a single FPLC, of course, but much, much smaller than 24 of them. It is a self-contained system, though, so the only exception is it has a computer that controls it that's separate, but there's no vacuum or air pressure needed, anything like that. It just runs on electricity. When it comes to lab placement, it is helpful to be able to easily access both the front and the back of the instrument in order to gain access to the pumps as necessary. If you don't have an open lab like this one, getting a bench that has wheels so it can be easily pulled away from the wall can be helpful as well. We also provide optional custom shelving and racks that can provide solutions for organizing samples in buffers. Here's a picture of how we often organize our protein samples at Emerald using 50 ml conical vials and this rack to organize the sample lines. And here's a picture of a shelf that several of our customers use that fits nicely on top of the protein maker and can easily hold 24 1-liter sample bottles or big multi-liter buffer bottles. We also make customized side shelves in order to suit your specific needs, and this is an example of one of those custom jobs. So now you have a sense of some of the specifics about the hardware, so I think it makes sense at this point to show you a demonstration method with the protein maker. This is a simple two-step purification method, but it will give you a sense of how the instrument actually operates. So I think we'll start by getting into the software and showing you how you would actually start the run. So this is the opening panel of the software when you open it up, and you can see a few different methods here listed in the list, and I'm going to select this two-fraction demo method. Within the method there is what we call the method tree, and it gives you different options for labeling and creating different steps within your purification method. So now that we've hit play, the machine will start running, and it will follow the order of commands that's listed in the method. The first action of the method is for the gantry to move to the waste position where the protein load step will take place. And what we'll do is we'll watch the instrument as it runs through this entire method, and we'll have this picture-in-picture of the software screenshot show up from time to time to give you a sense of where we're at in the method. So right now the protein load step is taking place. You notice there's only two 1 milliliter columns loaded on the gantry. We could have loaded 24 columns, but for the purposes of the demo experiment, we've just loaded the 2 columns, although all 24 lines will be active throughout this run. So right now the protein is being loaded on. There's an aspiration step where it loads, I think, 1 milliliter of sample and then pushes that 1 milliliter sample over the column. The next step in the method is for the columns to be washed. So what will happen is the gantry is now moving from the waste position to position C2, which is the position just behind that plate labeled F1. And we'll go ahead and wash the columns with a small amount of buffer and collect that wash in the 24 well plate. Again, all 24 of the lines will be active here. And the reason that all 24 lines will be active is because we have defined it that way within the method. So the software allows you to group the different channels together into groups of 4 or 10 or 1 or all 24. In the case of this method, we've grouped all 24 syringes together. We call that a column group, and that's what's highlighted in this screenshot here that you see on the screen. So what we've done is expanded the view so you can see a little bit more of the detail and some of the actions involved in this wash step. You can see where we've defined the column group as a custom group that we created before starting the method. Now the first wash is complete, so the gantry will move from the position C2, where the first wash was collected, over to position C3. While this second wash is taking place, I'm going to show you a little bit more detail about how to create column channel groups and just how that works. So there's an instrument setup tab within the software, and in that setup tab there's a place to create custom column channel groups, and that's what we've done for this method. We've created a column group that uses all of the syringes on the instrument. But you can, just by clicking these little icons, you can remove particular syringes from the grouping. So this group, for example, would have 16 syringes or 16 columns used. But you can select any number from 0 actually up to 24. You can group them in any way that you want. You can also custom name the valve positions so that they show up within your method in a way that makes sense to you. So for example, protein sample or elution buffer or tris buffer, you can name them whatever it is that you prefer. So that's how you create custom column groups or other flexible and customizable things about the software that we won't get into in this video. But now our second wash is complete, so it's time to elute our protein into the 2 fractions into the plates labeled F1 and F2. So we have a 2-step gradient that we're going to do, and the first step will be at 50% elution buffer. So how do we create that 50% solution? If we go up and watch the syringes, here's how it works. First we pull in 0.5 milliliters of elution buffer. Then the syringes pause, and then they pull in another 0.5 milliliters of equilibration buffer. Now we have a 50% solution of elution buffer. To make sure it's well mixed, the syringes will push that 1 milliliter of solution up into that passive or step mixing syringe that I mentioned before, and then pull it back into the glass syringe again. This mixes that solution very well and lets us know that we have a 50% solution of elution buffer. Other concentrations of elution buffers can be made in much the same way by pulling in different amounts of elution buffer and combining with different amounts of equilibration buffer. Using this mechanism, then we can create 2 or 3 or 4 or 10 step concentration gradients using that step or passive mixer. Of course, if you don't need to make any gradients at all, you can remove that passive mixer and use that port for a whole different buffer. Again, the system is flexible from that standpoint. So we've collected the first fraction at 50% elution buffer. The second fraction is going to be collected at 100% elution buffer, so there's no mixing needed in this case. We'll simply aspirate elution buffer and then dispense it out over the column. While this fraction is being collected, I'll just point out a couple other features or ways in which the protein maker is flexible to your needs. You notice this fraction is obviously being collected in glass tubes that are in an SBS format. It's a different Z height there, but that can simply be programmed into the method. So you can collect really in a variety of different types of receptacles. There are many different parameters within the method that can be customized. For example, aspirate rates, dispense rates can all be grouped together or controlled individually. Each line could have a different dispense rate if that was necessary. I mentioned before the concept of global parameters. It was one of the things that we saw in the little method wizard before we started the run. Global parameters are something that you, as the designer of the method, can decide to enact or not enact. It basically gives the power user of the software some control over what gets to be edited and what doesn't get to be edited. So you can define parameters as global parameters and allow anyone who's running the method to change those parameters at the last minute. For example, you might want to change the protein load volume. Maybe this time you have 50 milliliters, but next time you know you'll have 75 milliliters. So you don't want to have to go through the method and find every place where that parameter comes up. Instead, you define it as a global parameter. You don't really have to go into the method editor. You simply edit that parameter just before you start the run. In general, the Protein Maker software is very easy to use. It's intuitive. It's been designed to give the user control over all aspects of the Protein Maker, to give you the most flexibility for designing your methods as possible. If you'd like to see more about the Protein Maker software, we'd be more than happy to provide a web demonstration. Feel free to contact us. We'll set that up at your soonest convenience. Well, you can see that the gantry has made a move back over to the waste position, and it's now finishing up the final step of the method, which is the column re-equilibration, an automated step so that the columns are ready to go for the next method. And that will bring us to the end of our demonstration. If you have any questions at all, feel free to contact us at any time. We'd be happy to answer your questions or tell you more. And thanks for taking the time to check out the Protein Maker.